Hi, and welcome to the Digital Research Infrastructure webinar series. This series will showcase digital research infrastructure projects and communities in the UK and beyond. Join our series to learn about state-of-the-art initiatives that aim to advance research tools and techniques, foster collaboration and data sharing, and accelerate discoveries across a range of academic disciplines and sectors. So we are your hosts at the Digital Curation Centre. A few of you may know us as the DCC with our lovely orange logo in the corner there. So we are a team that's based in Edinburgh. We've been running since 2004, based in Scotland, in Edinburgh, as I say. Our mission is to help organizations worldwide make the best use of data in and for research, development, and operations in all contexts, be that academic, industrial, governmental, and more. Our team provides expert advice and practical assistance in storing, managing, protecting, and sharing data and obtaining the best value from it. Our services range and include hands-on training, tailored consultancy, expert data management tools, including our DMP Online platform, our International Journal of Digital Curation, our IJDC journal, as well as our annual International Digital Curation Conference, actually finishing last month, I should say, in Zagreb, Croatia. We're hoping to announce our next year destination soon, so do keep an eye on our social media channels for that news as well. But before I introduce our key speakers today, BioFAIR, I just want to find out a little bit about yourselves and who's joining us on the call today and where you're calling in from. So I'll do that by hosting you on WooClap. So it's a nice, easy tool to use if you are using a handheld device, an iPad or a phone. You can simply copy the QR code on the screen, or if you're using a browser, you can simply type in wooclap.com and then enter the event code RSPIGE. I can see a lot of you are familiar with this tool, having already had 17 of you join in. So we'll just be asking you a couple of questions about who you are, where you're calling from, and your familiarity with our speakers today before I hand over to Tony Burdett and Nishadi Silva to tell you a bit more about BioFAIR. So with that in mind, we always start this call by asking what country you're calling in from. Whilst this is a series predominantly about the UK, we are speaking about people who are further afield as well. So with that in mind, we already have someone calling from Saudi Arabia and Denmark, adding to a few members of the UK as well. We always have a good representation from the UK, given our current topics, but it is good to see people outside of that space as well joining us too. So again, I'll just leave this up for another 30 seconds or so you can tell us where you are calling in from as well. So again, welcome to those who just joined us for the second DRI webinar on BioFAIR. This is just a quick WooClap poll to get to know who is calling in and where you're calling in from. So now that we've found out where you're calling in from, what is your job title? This helps us to kind of get a bit of context around the kind of types of jobs that are joining us today. So as well as where you're coming from, to understand what it is you're doing day to day. Always good to know. So helps our speakers set the context of their presentations as well. So I certainly can see a lot of research positions there, some technical officers, project managers, computational biologists, as well as a learning officer as well. So it's good to see some variety. We always have a strong following in research, of course, being data specialists ourselves. But it's good to see other positions in there as well, and particularly with our leaning on the life sciences and physical sciences last month with PSDI, some more people from that background as well, which is always good to see. I'll just get another 10 seconds on that just to tell us who you are. So we even have an apprentice as well. That's great. So people of all levels, different industries and backgrounds as well. So it's good to see you all coming in. Thank you for joining us again today. So I'll just move on to our next question. So are you familiar with the concept of FAIR? So this is a closed question, just to give us an idea of your understanding. A nice easy one to tick off. And again, it's great to see so many of you are familiar with the acronym. This will be explained in further detail. by our colleagues at BioFAIR as we go through the call. But again, welcome to those who've just joined in again. This is a couple of questions before I introduce our speakers from BioFAIR, who will tell them a bit more about who they are. So a strong knowledge of the subject, which is really good. So certainly a great sense of familiarity with that. So just before I introduce BioFAIR, let's ask the question of how familiar you are with the team and the organization as well. So always good to know. Oh, great, not at all. That's always good. So you're on the right call. And even those who are very familiar and know a little bit about them, you'll certainly know more by the end of this call as well. Okay, so we got a bit of a mix of, sort of 50-50 split on not knowing BioFAIR yet, and knowing a bit about them, and a few who are certainly familiar with them as well, which is always good to see. So with that in mind, I will stop talking, I'll stop sharing, and I'll hand you over to the team. So we've got Tony Burdett, who is the director of BioFAIR with us today, and his colleague Nishadi de Silva, a senior project manager. Welcome.
Thanks. All right, let me just share my slides. Hopefully everyone can see my screen now. I think I'm
Yep.
working. Yep, great. Okay. Good. Thank you. Well, yeah, thanks for the introduction and actually useful readout on levels of fair awareness. Um, I don't have to explain fair is what I've taken away for the most part. But I do have to explain BioFAIR. So this is, I think it's a good opportunity to talk about BioFAIR to this group, and thank you for the opportunity to speak. Um, so just I see some familiar names on the call, but just I thought I'd start with a brief introduction about myself. For those of you that don't know me, so I'm the director of BioFAIR, which I'll explain exactly what we do in a minute. Previously, I was the Elixir interoperability platform lead. I haven't updated this slide yet. I rolled over at the end of last year and handed over my responsibilities. So Elixir is the European Life Sciences infrastructure, and my role there was to lead the interoperability platform, which had quite a strong focus on interoperability between tools and fair data management and data principles as well. I was also previously a group leader at the European Bioinformatics Institute for those of you that know that organisation. So my background is fairly heavily focused on building services and working on fair data management in the context of life sciences and bioinformatics. My background originally I've got an undergraduate degree in biology. I actually spent some time at medical school as well. My master's is in bioinformatics. But broadly speaking, I built and helped develop or run the development teams for lots of major life sciences data resources. There's a little selection of them there. Lots of interoperability tools, but really sort of resources that support the research process with data in the life sciences. That sort of my background is sort of, I guess I was sort of a research software engineer for a while, in a sense, not really involved in frontline research, but more developing the sort of resources that support that process. So, sort of probably a similar career trajectory to a lot of people on this call. Um, in March last year, I came on board as the director of BioFAIR, and BioFAIR is still a relatively new federated digital research infrastructure. The idea is to provide a sort of step change in FAIR data management for the life sciences. So we're really targeting the idea of providing end-to-end research data management capability, both in terms of data analysis and data storage, data support, along with the training and support that's needed for UK researchers working in the life sciences. The training, the people angle is a big piece of the presentation here, is a big part of what we're doing. We're a initially 5-year funded initiative, backed by £34 million of UKRI money. So we've had quite a large investment to create a sort of culture change in pan UK life sciences research. So It's quite an ambitious sort of undertaking to do this across the whole of the whole of the UK for all of the life sciences domain. But we're lucky enough to have been given lots of backing from UKRI to achieve that. Um, I thought just to start with, I should sort of highlight what we think about as digital research infrastructure. Hopefully, with this group, there's a decent idea of what we mean. But what we tend to find is people use digital research infrastructure to mean lots of different things, all the way from disk compute, the sort of hardware, all the way up to research data management support. And we think about this in BioFAIR as all of those, and in a sort of very federated way. So our interpretation of digital research infrastructure on the right hand side here is we need to do four things pretty well. We need to store data securely. We need to give people access to computing power to do research, access to shared tools so that they can use them, and also really facilitate the easy collaboration. And those things we think are the things that are needed to empower life sciences research. BioFAIR being a federated um, digital research infrastructure, we designed it that way because there are loads of services already out there, services, systems, support already out there in the UK life sciences. Lots of investment has gone into this space in the UK over a long period of time, but in a somewhat fragmented way. Which means there's all the pieces are there, but they're not necessarily assembled in the best way. So we think about BioFAIR's digital research infrastructure as having this role of like picking up and dropping in all of the pieces that we need, but weaving them together into a sort of fabric that brings them to bear more efficiently on research. So we very much think about the things that we're trying to do with BioFAIR as providing the connective tissue or the glue that links together these things on this slide. So thinking about what data repositories are well used in the space, what compute engines that we need, what workflow orchestration tools, what API standards, what sorts of interoperability formats are needed, and the user support. These things all exist, but we just need to drop them into the right places and assemble a sort of overarching fabric that enables life sciences research to happen more effectively. Based on the introductory poll, I don't think I need to explain this to this audience, but we very much think about how important fair data is. And this slide is a little bit old. We're actually trying to commission some work to update it. So the article that sources this data is more than 10 years old now. But we know, I think anecdotally we know, and we know from data, and every time we ask the question, that data science and digital research is heavily bottlenecked by the availability of good data. So actually, data scientists spend 80% of their time collecting and cleaning data, and often it's the part that they enjoy the least. Not everyone is. It's not true of everyone, but I think especially in an era where we're embracing AI is based on the idea of having really good data, or it succeeds with really good data. The impact can grow if you've got good, well-organized data to feed into machine learning models and algorithms. And the sort of current level of or the current effort that goes into well organized, clean, well structured, fair data doesn't really support that level of reuse. You see pockets where it works really well and other places where it doesn't, just because the availability of the data is not there. So really what we're trying to do with BioFAIR, one of the things that, one of the ways we think about our success is streamlining that process, streamlining that 80% of time spent just cleaning and collecting data sets through sort of four main pillars: maximizing the fair and reproducible data and methods through targeted ways to improve data management culture, mobilize researchers to share their data in a more fair way, for example, reducing the waste time, the amount of time spent trying to go recurate data sets, going back to publications, going back to source data just to try and clean it up so you can use it, and improve the AI readiness of data. But then also through widening the use of methods and services, not just the data, but actually thinking about how to optimize best practice around reusing tools, reusing workflows, gain support from people that have run those tools or run those workflows before, benefit from international collaborations. these sorts of things. So really thinking about broadening the use of and reuse of existing tools and methods, so it's easier for researchers to find and reuse the tools and the data that they need. That we think that makes it easier and streamlines collaboration. So if you're working on a shared problem with shared data and shared tools, it's easier to collaborate. Um, and also thinking really quite carefully about how to bridge the gap between researchers that are driving primarily driven by an approach of identifying new findings, new knowledge, new outcomes, new insight, and bridging that gap between the researchers and the data sources that are hosted at individual institutions or data infrastructures that are available to them. We find that there's a gap, and I'll talk a little bit more about that at the moment. And sort of one of the success criteria for us there is thinking about how to increase the efficiency of research through bridging the gap between infrastructures that are available and the researchers that use them. So our vision is really then for the UK's digital infrastructure to put the answers to life sciences questions at researchers' fingertips through simple, efficient reuse of data and methods and expertise of potential collaborators. And this is intentionally broad. When we talk about the UK's digital infrastructure, I think it's not just the stuff that we're doing in BioFAIR. The answers to life sciences questions aren't siloed out into sort of purely life sciences data repositories or methods. They run across domains. So there's loads of instances where we're looking for what's the impact of climate change on human health, or what's the impact of climate change on crop yields, or these sorts of things. And so that it's intentionally a sort of cross domain view on the world, really thinking about how we answer research questions in the life sciences as simply and easily as possible, reusing data and reusing methods wherever we can, and collaborating with people who can help. And that gives us 5 quite specific objectives. The top one, and the beginning and the end of our problem really, is about culture change, driving adoption of FAIR principles and open data across the life sciences. We think about 3 sort of ways of achieving that. One is defragmentation. We want to increase the coordination, collaboration, and cohesiveness of the life sciences related data landscape. And that's very much thinking about this pick up and drag in and drop pieces of work that have happened elsewhere. So that enhances its efficiency. We want to think about democratizing access to data and methods within the UK life sciences through sort of a national capability. So rather than things being stuck within individual institutions or very localized within niche communities, to then give them a national presence and national capability. And then to attract, develop, and retain excellent research data management skills. So this is really about the people and sort of magnifying and amplifying the skills of really highly talented people in this space, and also bringing people into that fold and giving them the opportunity to train and learn new skills. And we think if we do all those things right, the culture change is going to promote increased data reuse. So the start and the end is culture change and data reuse. I think those 2 things are interconnected very intimately. Now, I did want to, so that's our sort of broad overview of BioFAIR. I did want to give a little bit of a flavour of some of the complexity of the challenge of doing this in the life sciences, which might sort of, I'm sure there are similarities, but there might also be a few little differences to different domains. So when we talk about the life sciences, primarily with BioFAIR, we're talking about open data. So that's not hospital records or clinical data sets from humans, from patients that are quite often locked away into hospitals or specific research programs. But we do think about more open data sets. So some of those open data sets may come from humans if they're sort of post-mortem samples or patients that are deceased, or historical records or cell lines that have been anonymized. But also it includes all of the other species that aren't just humans. So plants, animals, anything that's sort of open is within the domain for BioFAIR, but it also runs across the breadth of complexity within the life sciences. And I've presented one view on this here. So one way of thinking about this is that all of the different areas of life sciences that explode in complexity. If you start from the genome, you've got 20,000 genes in a human. They're that's the raw DNA component. As you move up and get closer towards an individual or a person or a plant, this gets more and more complicated. So you move from 20,000 genes in a genome to 100,000 different types of RNA transcripts. So you get these kind of phenomenons where one gene is transcribed in different ways. You get alternate transcription sites or different ways of splicing it. So you get a sort of explosion of complexity from 20,000 genes to 100,000 transcripts. Those things, that RNA then gets turned into proteins, and there's a million different proteoforms in a human, roughly speaking. And that's, again, lots of different ways of creating a protein or folding a protein. Those proteins interact with each other, and we call that the interactome. And ultimately, that creates a sort of set of functions that typically is called a phenotype. And there's areas of study that go from genotype to phenotype. So if you're interested in one particular disease and maybe the genetic basis of that disease, you're going to think about all the spectrum of this genome to phenotype for one particular disease. Or there's some things that start with a gene and go the other way, and there's all sorts of different ways of slicing this problem. But you get this problem of exploding complexity when you get up to whole species or systems or interactions. It's a really complicated field to study. And what that means in the life sciences is that you get these kind of many different fields of studies that we then try to group back again. So Elixir's done that by thinking about communities. There's multiple different communities in the Elixir, the European Life Sciences infrastructure here. So, you see there's like marine metagenomics or metabolomics, plant science, single cell omics, these sort of natural groupings of like common areas of practice. It's not just one way of doing life sciences, there's lots of different ways. And we try to sort of cluster and group them together. Australia BioCommons, that's kind of the BioFAIR sibling in Australia. They're a bit ahead of us, and we've been learning a lot from them. They do the same thing. They group into domains, and like you see some of the same ones here. So there's genomics, genome informatics, metabolomics, single cell, these sorts of things. EMBL EBI also think about the world in a similar sort of way. This goes from genes up to systems. So bioinformatics runs across all of these areas, different fields of study. Some run the whole arrow or the whole spectrum for one particular disease or one particular condition. Some just look at one particular type of study. Some are more computational. Some are much more wet lab. So there's many fields of study, and there's many resources to support those different fields of study. This is the 52 global core biodata resources that are sort of characterized by the Global Biodata Coalition. But there are hundreds of thousands of these things when you get down to like quite fine-grained sort of distinctions within universities, within organizations, within communities. These are the sort of big global 52 that are considered to be the resources that if they went away, they'd have a sort of catastrophic impact on a particular area of life sciences research. But I think, and you can appreciate the sort of size of scale of complexity of the life sciences that we're sort of trying to sort of integrate back together and identify interoperability for. So that gives us a FAIR challenge, right? And I think we know that all of us here know that adoption of FAIR is hard, and changing culture is a lot more than knowing how to do FAIR, but it's actually, where do I start? And we think about this in BioFAIR with this kind of how fair is fair enough idea. And intuitively, I think we all know, I think everybody on this call would recognize this kind of dichotomy that starts at one side of the world is use data once and then throw it away, and on the other side of the world you've got data reusable for every possible use case now and in future. And often we end up sort of having some sort of discussion about the pendulum swing here, and everyone knows that this is a sort of false dichotomy. Both of these two scenarios are ridiculous, right? You don't want to be stuck in a situation where you're collecting lots of really good data, doing a deep research program, and then nobody ever can use it ever again. And you don't want to have to think so long and so hard about how to make your data reusable for all possible future uses that you don't have the time or the energy or the money to finish your research program. So, I like to talk about this as being a spectrum of fair rather than a sort of, you can either be fair or you're not. And then you can start to think about nice things like value for money, right? So I think everyone knows that there's a good level of work that you can do that will accelerate your research program, and support people collaborating with you, but perhaps you don't want to be all the way over in this data reuse for every possible future use scenario. So this fair is fair enough, right? This kind of point in the middle. The trick is identifying exactly what that looks like. And once you can start to do that and think about what you're trying to get to, what actually is my goal? Where do I realize and benefit? This is where the culture change component comes in. I think that many people are scared of starting a FAIR journey or progressing on a FAIR journey because they're worried about the potential costs of making their data reusable for every possible use case. They think they're going to lose out on the value for money curve. If you can highlight and identify where here is, then you've got a really positive way of creating culture change. So we've spent a lot of time thinking about this. And one of the projects I've been involved in previously before starting in BioFAIR was the FAIR Plus project. This ran from January 2019 to December 2022. So right through the pandemic, doing this project through the pandemic was fun. But we really spent a lot of time and energy thinking about data from the Innovative Medicines Initiative project portfolio. This is translational research, so projects that look at can we develop new drugs that treat human disease. The IMI program had been running for a long period of time by the time FAIR Plus was funded. And what they'd found was that more than 100 projects that they'd invested in, much of the data never got reused. And so the FAIR Plus project was funded to say, well, actually, how do we improve this? How do we design, test, refine tools and processes in a way that scales to the volumes of data that we're talking about? There were some outcomes, but I want to talk through one aspect of this that we found that I thought is kind of relevant to this kind of FAIR problem, and we're carrying forward or trying to carry forward into BioFAIR. So essentially, what we found was that data is not reusable at scale, and that's what we tried to support with FAIR Plus. And we broke this down into four main challenges. One, there's no practical advice for people on how to do verification. That has improved a little bit since the days we were involved here, since 2022. We worked on a FAIR cookbook. There's also other tools and services now that provide guidance for verification. We found that people couldn't tell how fair they were already. So we spent some time thinking about how to assess how fair you were, assess your fair levels. We found people didn't know how to become more fair, which we had a set of processes and a maturity model that helped. And we found that organizations don't care anyway, which isn't always true, but sometimes it is. And that's really about changing the data management culture. And that speaks to the sort of curve of cost benefit. And that value for money curve that I just showed. I won't spend a lot of time on this, and you don't need to read this slide, but what we did through FAIR Plus was identify a sort of verification process that started and ended with defining a verification goal. This is really about how fair is fair enough. And then, like many people here, I think will sort of feel and know intuitively from their working experience, it then involves a series of experts understanding business objectives. What's the verification goal? And then working iteratively and incrementally to improve things through cycles. That's what the cycle on this diagram shows. And importantly, what we found is it's really important to disseminate your learnings once you get to where you were hoping to be. Because people often don't do this. They just sort of keep going until they're experts or sort of drop away and never share any of their lessons learned. And we came up with this framework that sort of formalizes this a little bit. And I won't go through this in a lot of detail, but there's lots of common things that people will recognize about doing domain modeling and choosing appropriate vocabularies and selecting identifier schemes. And also thinking about where to host and share the data. And these things are sort of then carrying forward into a lot of what we're doing now with BioFAIR. We came up with a template for verification. If you're interested in this, I would encourage you to go read the paper that we wrote up. I'm sort of sharing it as a sort of highlighting of like this challenge of what we found in FAIR Plus and what we find in BioFAIR as well is that often the problem is not about knowing what to do, but knowing where to start. What should I do next? How do I progress? What will I get back from it? And that was the core behind this verification framework. And the other thing I think that we've learned and built on in BioFAIR, learning from those kind of experiences is and I talked about trying to bridge the gap earlier. What we found and what we still find is that there are sort of broadly speaking three sort of sets of or three audiences for BioFAIR. There is researchers who are primarily motivated by getting a paper out, making new insight in the life sciences domain. There are digital infrastructure providers who are building databases or building workflows or building workflow systems, or tools or services that support that research process. But most of the link between those two is held up by research technical professionals who are then bringing their specific technical expertise about how to be fair, how to use services, how to define vocabularies, how to do ontology mappings, all these sorts of things, and actually actively supporting researchers. And what we find is there's not a clear bridge there, but the research technical professionals are often holding it together, like either manually or bits of string or holding up like planks in a river, if you like, rather than a bridge to sort of support that whole process. And we want to try and change that with BioFAIR. Now, this side of the bridge is kind of users. I think BioFAIR will ultimately provide services to researchers and support for research technical professionals. Whereas digital infrastructure providers are more like BioFAIR partners. Research technical professionals are also our partners here as well because they can help define the approaches, the tools, the methodologies that BioFAIR is sort of building on and then making more available to researchers. So that's a little bit about how we think about the what we're trying to achieve, how we're trying to achieve the culture change and the people that can help us do it. In practice, we sort of think about three main pillars of BioFAIR, which we've turned into our BioCommons. There's a data commons piece, which is really about enabling researchers to prepare, analyze, share, and manage fair research data, and importantly across that whole life cycle. So right from the point of generation to the point of publication. The methods commons, which is about enabling institutes and research teams to access trusted and robust analysis platforms, so take advantage of existing workflows and be able to run them in one click, run a workflow that someone else has tried and tested or is the best practice for your domain. But they're the bottom layer to the people. And I think the people commons that BioFAIR is trying to create is really the important bit. We're thinking about a pooled community of data stewards, research software engineers, sharing practices, sharing knowledge, supporting data and software management planning, and really making that sort of those technical pieces possible for the research community to use through that sort of direct support function that I imagine many people on this call today already play. And we sort of break that down a little bit further into what we call our structural model. So the BioFAIR hub is at the center of this. That's where me and Nishadi are based. And surrounding that is sort of a federated network of what we call our commons layer. So data methods people is in here. We think our knowledge center fits into that layer as well as a sort of data portal, which at the minute is quite basic. It's just our website, but we hope to build on and expand out over time. The green layer on this diagram is then service providers that we're sort of picking up, and this is the pick up and drop in approach. We know that there's loads of services out there that we want to integrate into our commons, whether that is data services, method services, training courses, people through our fellowship program. There's loads of stuff that we can sort of integrate and weave in together. And then round the outside of this, we think about the platform offering a concierge service. So that's really about thinking, how do we bring those people along? How do we bring researchers into the fold, teach them how to use services, make it as easy as possible for them to take advantage of the commons and the services that are there, and a little bit of targeted investment to build that out through our Pathfinder projects, which we think of as sort of our early adoption users. And we're really hoping to create that culture change through thinking about growing alignment and a sense of mission. And that's between teams that have complementary skill sets that sometimes don't speak to each other in the current setups because they're too siloed out or there's not appropriate forums for them to share their knowledge. So creating a sense of alignment, a shared sense of mission, increasing collaboration, especially between research technical professionals, and enhancing knowledge sharing around tools and techniques for best practices, excellent data-driven research, and then driving awareness and driving efficiency in the research process itself through reductions in reinventions of the wheel. So that's the sort of things that we're trying to change. We've got 6 kind of current targets on and this is now zooming into kind of what we're doing quite currently. At the minute, we're spending a lot of time working with community of users and stakeholders to evaluate needs and capture requirements. What would the BioFAIR platform do for you? We're building out a sort of analysis platform, and we're actually just identifying a new set of delivery partners for the methods commons to help us develop the technical services for data analysis workflow execution. We've started building out a community and building out champions within that community. So we've got BioFAIR fellows, for example, that are supporting researchers across the UK and building out this pan-UK community to act as ambassadors for BioFAIR. Later this year, we'll move into actively sharing data for analysis more carefully, building out a concierge service that I've just talked about, and also establishing all the core operating framework, the network in the hub that we've been working on for a while. So that's sort of really what we're doing and how we're approaching things. I also wanted to share a few thoughts for the future, and we're including a vision for BioFAIR and how we're sort of trying to think differently about this kind of data science and biodata resource ecosystem. So I mentioned the BioCommons already. As this sort of the way that we're trying to approach this in sort of 3 main pillars. If you zoom in this a little bit, really we're thinking then about joining up the dots. So that we know that there's kind of this world of existing data, tools, and resources. There's some things mentioned on here that's by no means comprehensive. But we're thinking about the BioFAIR portal as being the thing that can bring life sciences researchers into the fold. That exposes a view over the data commons, methods commons, people commons, and also sort of makes knowledge available. And in turn integrates institutional repositories, community repositories, and registries, as well as all the tools and services. And in this way, BioFAIR sort of joins the dots. It connects DRI investments, acts as catalysts for integration between different data hubs, both within the life sciences and across the life sciences. So if you have thoughts or insight about how we can collaborate, I'm really keen to hear from you. And we're also sort of emerging a BioFAIR technical platform. And this is just a sort of very high level view of the sorts of ways that we're thinking about our infrastructure. Obviously, our researchers want to interact with the website through the portal. And there's sort of 2 different ways that researchers will interact with our services. One is in researchers who are actively trying to do reproducible research that'll come in through the website and sort of use the services that we have in the technical pieces. So this is the Knowledge Hub, Data Commons, Methods Commons, and we kind of think about the interactions for researchers doing reproducible research as following these kind of 3 phases that hit those commons pieces in turn. Firstly, you sort of need to set up a good data management plan. You need to understand how to manage your data, how to be fair. And resources like DMP Online or Fair Sharing RDM Kit really help you with that process of like that setup phase. I've just kicked off a new project. I want to figure out how to do a data management plan. What do I need to do? That sort of flows through. Once you've got your data management plan, that flows through into how do I manage my data from the moment I start collecting it. And that might include things like global core biodata resources, or it might just include institutional storage, like it might be an iRODS, something like this, or Amazon S3 or Zenodo. Managing the data as you're generating it and then flowing through into how do I actually analyze my data in reusable ways? And the 3 sort of technical components in the middle of BioFAIR, this middle of this BioFAIR sandwich here, are the things that really drive reproducible research. But if you can do that, then the outcomes that you get, the research objects that you get out at the end, the research outputs, are then things that you can link from peer-reviewed journals and that other researchers can reuse really effectively. So we're really thinking about our technical platform that's emerging is really thinking about 2 sets of research users, those trying to do reproducible research and those trying to reuse research outputs. But that's not to forget the research technical professionals who we think might interact sort of in a slightly lower level and help us with building out a sort of really robust data stewardship toolkit. Now, yes, they can provide components into the technical platform, but actually the tools that make it easy to take advantage of these things, like like DMP Online, for example, is one of those. We might have like metadata validation tools or ontology lookup tools, these sorts of things. Can weave a really important part into the overall BioFAIR platform as well, and through that way we bring really effective research support tooling to research technical professionals. We also anticipate the need to have like a foundational platform that makes commercial cloud and HPC and AI compute facilities available to the research community, and we're sort of starting some conversations about that. I did want to offer the obligatory thought for the future on our approach to AI readiness. And I've talked a lot about fair data in this presentation, but I really think fair data is at the center of this kind of onion for AI readiness. It's only one piece. You need fair data. It is important, but you need a lot of other stuff on top of and around the fair data itself. So you need digital infrastructure that can support AI experimentation and AI production. So both making it possible for people to try out AI, is this going to help me with my problem? Can I build a workflow using AI that might design, or gather new insight? But also, once I've done that, how do I turn this into a reusable method? We need software and tools, I think, that are accessible, well tested, and robust, as well as interoperable, that take advantage of the data and the infrastructure so that you can start to sort of train models up or take advantage of models that already exist. And then ultimately this rolls into this kind of on the outside layer, this reusable models, and both the models themselves and patterns of usage that work well. So, is an LLM good for my particular type of problem, or do I need a RAG or something else? And organizing those by the types of problems and looking for learnings that transfer across domains, I think, is really important as well. BioFAIR, we're trying to do this in four particular ways. We're trying to be very team first, so very focused on sort of building out teams and making sure we've got teams with the capabilities to help us work through this set of challenges. We're creating with the community. As I've talked about a lot, we're using existing services and delivery partners, and we're also trying to think a little bit differently about how to create this culture change. Why has it not worked in the past? And I want to talk a little bit more about that. I have got a few slides that I'm going to sort of skip through in the interest of time. We do want to create with the community. We're trying to learn from previous initiatives on this, like Elixir. Elixir's done a very good job of like building community engagement, and we have close links with Elixir and Elixir UK that we want to take advantage of, so that we're not reinventing the wheel here or trying to create yet another view on the same sets of communities for create more meetings for people to attend. But we do want to do this kind of co-creation. So, we're not trying to design something in an ivory tower. I've talked extensively about this already. We want to make sure we're using existing services and delivery plans for the methods commons and the data commons. There's some examples on this slide, but thinking about the capabilities that we need as well. So do we need workflow analysis capabilities for Nextflow and Galaxy, or do we need sets of standards? We know these are the things that we need, and we've defined exactly what we need in the methods commons so far. We're still scoping out a little bit for data commons. So if you've got sort of thoughts and questions and you're interested in that space, please do follow up with us. And especially think about sort of things like knowledge graphs for data discovery. Knowledge Hub and People Commons. The People Commons is already well underway. We've got recruited a set of fellows. This year we'll be thinking about training programs and more community engagement. So you might hear more from us in that People Commons sense. And one of the initiatives that we hope to launch this year is the Knowledge Hub component, which might include sets of standards registries, training portals, RDM toolkits, and there's just some examples here of like FAIR Cookbook, RDM kit, FAIR sharing that already provide some of these sorts of services. I did want to talk about Think Differently just in the last couple of minutes. So, right at the start, I highlighted these 52 global biodata coalition resources that are really important. But we also know, and this sort of relatively well understood problem of data journeys, that this diagram on the right hand side of this slide is from the RDM kit. Planning, collecting, analyzing data all the way through to data reuse. And I often think we have this sort of fundamental sort of split between think about data resources or think about data journeys. And I like to summarize that with this architecture diagram. Every data resource in the life sciences, every biodata resource ever thinks about the world like this. It has an architecture diagram. That's a great big fat database in the middle, some tools that are available to data submitters, and some tools that are available to data researchers. And maybe if you're lucky, you get one or two little tools that sort of take data out of the database and put it back in. But fundamentally, it's this very centralized model. There's a big database in the middle and tools for submitters and tools for researchers to put data in and get it out again. I have a problem with this because I don't know who is responsible for the usefulness of the fairness of the data in this world, and it seems to get passed around like a hot potato. So you get curators who are sat within the biodata resources for a little while, and then that group gets downsized, and you try and pass the responsibility on to submitters, and then they don't like to do it because they're just trying to get their publication out. Or you try and pass the problem on to researchers and say, well, we're a generalist resource. We have very open standards, and you can recurate from everything's in there. You can recurate from the literature if you need to. And we since have this problem of fairness being a hot potato. I highlighted this program problem as our key audience earlier on about researchers, research technical professionals, and digital infrastructure providers needing to work effectively together rather than sort of research technical professionals being thought about as sort of a second class citizen in that model of like, we've got two sorts of users, we're the digital infrastructure provider, and we try and pass the problem around of who's responsible for the data fairness that then ends up being done by research technical professionals. What I like to think about is I'm going to bend an analogy to almost breaking point here, but this is a picture of a very typical sort of French tree-lined boulevard. If you've ever spent any time driving around France, you will have been down a road like this. There's great straight roads with beautiful like lines of trees on either side. The sort of urban legend, perhaps I don't know how true it is, but don't let it get in the way of a good story. Is that Napoleon planted these trees in war and in the Napoleonic Wars, and the sort of story behind this was that Napoleon knew that rather than trying to train his soldiers harder or have them carry more gear or bigger swords or whatever to try and win the battle, he was better off making sure that his soldiers could arrive at battle fit and healthy and ready. And he had the insight that by planting trees alongside all the roads in France, it signaled the route from A to B. It provided shade for his soldiers to walk in all day. It gave a ready supply of firewood, so when they camped overnight, they've got plenty of wood to make a fire with. Some of them got fruit on them, so there's food. And he knew that actually, if he changed the environment and made sure that his soldiers arrived fit and healthy at the battle, that was much more effective than having twice as many soldiers or giving them twice as much gear or whatever else. And I like this kind of insight because it's really sort of creative thinking outside the box to change the environment rather than just invest more heavily in this obvious thing. Change the environment about what you're doing. And we've been trying to think about doing that in BioFAIR as well. So rather than thinking about the resources themselves and how do we build the resources up, but how do we build better tools, better databases, instead try and change the environment. So I think we've been trying to think about the journey. So thinking about the use cases for data, encouraging people with data to think about use cases for that data. Why are you collecting that data? What data journey does it support? Try and map data as it goes along a journey rather than create a centralized resource, create shareable workflows, consider what resources might be stops on the journey. How do we chain things together? So try to make the journey easier rather than overloading already busy people or monolithic data platforms. So try not to create yet more silos. Don't create one database to rule them all. Stop reinventing standards. Stop stretching successful data resources beyond their usefulness. Better to do one thing well. So we've been trying to think about ways to achieve that. I will finish on that slide and take questions. But yeah, I think for the sort of moral for BioFAIR, I think is there's lots of opportunities to get involved right now. We're doing lots of things with our infrastructure in this slide, and we'd welcome collaboration and support from this group. And also, I think if you've got thoughts and ideas about this kind of data journey mentality of like integrating and then making it easier for data to flow between resources and arrive with researchers who want to reuse that data more effectively, rather than trying to overload existing data resources or build yet another data platform. Really interested to hear them. So I'll stop there. Thank you. I appreciate I went a bit long. Not too much time left for questions, but I'll stop and throw it open to the floor. Thank you.
Thank you very much, Tony. A very information-rich talk. So we do have a few questions in the chat. I will change the order a bit, and then we could have a little bit of a discussion if time allows. So Ding asked if BioFAIR is only currently aiming to serve researchers from the UK. So is the focus on the UK only? And what about the future? What about the international communities in life sciences?
Yeah. So yeah, I mean, our mission, we're funded by UKRI, so we're targeting researchers within the UK. I mean, I think we've been approaching this in a fairly international way. We've been quite collaborative with a lot of our sibling initiatives. So we spent a lot of time talking to Australia BioCommons, folks in Germany from NFDI, folks in Scandinavia, SciLife Lab, in the US as well. I was at the RDA, the International Data Week last year. So we're trying to do this in an international way, but our program is funded to support researchers in the UK. We're especially not funded to provide free compute to researchers outside the UK, for instance. So there's a bit of a balancing act. We're trying to do this in a very collaborative, very interoperable way, and thinking about how we adopt global standards. But our mission is UK focused. Yeah.
Yes, understood. Thank you. So another question from Kevin has to do with incentives. So the current academic publishing landscape is still putting publications, articles first, even if the data supporting those articles is not fair. So how do you see the role of BioFAIR in changing that aspect?
Yeah, I mean, there's carrot and there's stick at play here, right? So, I mean, I think we have, I mean, there's been already historically there's been initiatives to encourage publishers to do a better job of making sure that data is fair and reusable before it's published. And there are many databases in the life, or many journals in the life sciences that expect you to put your data into one of the global core biodata resources before you can publish. Um, and so there's a certain amount of fairness. It probably doesn't go very far enough, and you still find even things that have got like accession numbers from well-recognized databases. The data is not really reusable at the level that you would want it to be. And I know that myself from running one of those databases, European Nucleotide Archive. There's often very minimal metadata, but at least the data is available. It's better than nothing. So there's more incentives. I think there's more carrot and more stick that could be deployed here. I think the way BioFair is thinking about it is trying to make it much easier to do the right thing. So that provides some incentives, not enough incentives because you can still get away with not being fair. And we're thinking about how we involve publishers. The UKRI are being very supportive with data sharing policies and improving the way that they want research to share data through funding mechanisms. So, that's a little bit of sprinkling of carrot and stick. We can make it easier, I think, for people to share their data in the right way and conduct reproducible research. But I think there also needs to be a bit more stick about what happens if you don't do that. So, yeah, there is a lot more to do in that space,
Yeah.
right? So, there are things that we can do, but it's a perennial problem as well. The sort of incentives for
Yes.
doing reproducible research are often missing.
Absolutely. So perhaps a related question. So Karis is curious about how you develop your communities and to learn more about the BioFAIR Fellows program.
Yeah, I mean, we could talk. We could probably do a whole webinar on that. So, we launched our first fellows program in October last year. Was when our first BioFair fellows started. We have 10 fellows. They're funded at 20% of their time. So essentially they get 1 day a week to do community building, community engagement, work with their scientific communities to sort of figure out their needs and whether BioFair can help them. So that's we're really only just starting that journey. We're only sort of 6 months in to sort of building out our communities. We plan to do a bit of acceleration in that over the course of this year. Um, we're leaning on connections to Elixir communities and things as well already. Um, uh, we could spend a long time talking about that, Kara, so perhaps we should follow up afterwards. The fellow. So, I mean, I think the easy way to answer that question quickly, I guess, is that our fellowship program kicked off in October last year, and we will do a new round of fellow. We have 1 year turnover for fellows, so our plan is that we have 10 fellows this year. They will move at the end of this year, when they reach the end of their year's tenure in September, they'll rotate into like the alumni of our fellows program. So we have a once a fellow, always a fellow approach, and then we'll onboard a new set of fellows with a call for fellowships later this year and bring a second round in. So that helps us sort of build out a network of people that are well connected in the UK life sciences landscape at the minute. But we're also trying to build communities around that independently.
Yes, thank you very much. So I know there is a lot more to discuss. Hopefully people will have a chance to explore BioFAIR website, but I will close the Q&A now and hand over to Al, who has one more poll for us to finish. Thank you so much.
Thank you. Thanks, Agnes. I'll just share my screen quickly with you all. So thank you again for that really interesting talk around BioFAIR. So I think given that at the beginning of this, we asked you how familiar were you with BioFAIR, it's only fair now to ask you if you have a better understanding of BioFAIR. And given our excellent talk over the last hour, I'm sure that will be a resounding yes to this. I'm conscious as well that we had a few more questions as well, which is really good to see. So I'll be passing these on to our speakers, and we'll be getting back to you with answers on those questions as well. So if you do have more questions on BioFAIR, or if indeed you are unsure about what they do, do get in touch with the team and email us at info@dcc.ac.uk. Conscious of the time, this will remain open, this poll, so you can continue to tell us if you are more familiar with the services offered at BioFAIR by completing the yes, no, unsure. But I'll just close with a note to our next DRI webinar. So our next session is happening on the 15th of April next month, specializing and focusing on the museum data service. So for those of you unfamiliar with this service, it's an organization that aims to connect and share all the object records across all UK museums. The organization provides access to data drawn from UK collections, provides both a national overview of the collections through summaries, as well as offering object-level records as FAIR data to transform the way that researchers access museum collections. So you can join us then to understand more about the work at MDS and how their support can benefit your research. So I'd like to say thank you to Tony and to Nishadi for joining us today. Thanks to Agnes for hosting the Q&A. Please do continue to drop messages in the chat. When we do close that, please do email us in our thank you email that I'll be sending around probably next week. We'll share a link to the recording, as well as answering a few of your questions as well, and the slide deck itself. So anyone who did miss that, be that people who registered or colleagues that were unfortunate to miss out on this opportunity to learn more about BioFAIR, you can share that with them as well. So at this point, I'll close the session. Thank you again for joining.
Thank you.